So our talk today is by Raihaan Patel, and Raihaan is a graduate student in my lab. He's been with me since 2014 when he finished his undergraduate in, oh, I already forgot the name of the program, engineering science, because engineering is not enough and science isn't enough. You have to mash them together and create this super program at University of Toronto. He worked with me as an RA for a year after that, and then has gone on to do his master's degree in biological and biomedical engineering, and subsequently transferred to his PhD. And he's kind of seeing the finish line now, and I thought it would be a good time to share some of his data with everybody. So without much further, you might know actually Ray from a lot of the work he does helping people figure out their stats or their image processing or what have you. So it'd be nice for him to be able to share some of his work today. So thanks a lot, Ray, and I'll let you have it.
Cool. All right. Thank you, and thanks everybody for joining. So today I'll be talking to you about two projects that I've been working on throughout my PhD, which I have nicely titled Investigating Microstructural Variability in the Human Brain. So I'll start with a quick overview of the goals and general outline of the project and what it's trying to accomplish. Then I'll get into some background. I'll talk a bit about multimodal MRI, as well as a technique called non-negative matrix factorization, which factors heavily into my work. And then I'll get into the two projects that I've worked on. The first one being a look at hippocampus microstructure in healthy adults, and the second being a look at cortical microstructure in a healthy elderly population. So I state the goal of my PhD works thus far is to develop and apply analysis methods for integrating multimodal MRI data to investigate microstructural variability of the human brain in the context of healthy aging. So that's quite a few words, and I'll highlight a few which I think are really the key. It's integrating multimodal MRI data or multiple metrics. I'm looking at microstructural variability of the human brain, and the population of interest here is a healthy aging population. So I'm going to talk about two projects today. The first is a look at hippocampus microstructure in healthy adults using Human Connectome project data. And so this project really was my first project in my PhD, and it really serves as a methods development and sort of validation to develop a framework for analyzing multiple metrics at the same time. And then the idea is in my second project, I move on to work with actually a healthy elderly population, applying the methods and analysis developed in project one. And I will note that, so here I'm talking about multimodal MRI data, but I'm specifically focusing on microstructural metrics as well as healthy aging. Although a lot of the methods here, I think, if those aren't necessarily the types of data or the population you're interested in, I think you can still take some things from this presentation. So you could swap out microstructural for another type of information, whether it's, most notably MR information, but if it's functional or something else. And you could also swap out healthy aging for another population of interest. And I still think there's some general themes in terms of analysis that might be interesting for you in this presentation. So I'll start with an overview of multimodal MRI and why it's useful, why it's important. So in recent years, there's been a real proliferation of multimodal MRI, and it's becoming more and more common in large-scale data collection, which is a great thing. So multimodal MRI refers to the use of one or more contrasts or sequences in a given analysis. As an example here, we're looking at structural, diffusion, and functional data for a given subject from the Human Connectome project. So the benefit of having all this information as opposed to just one of them is that each MR modality and each MR metric can provide complementary information. So by looking at all of them together, you're getting a better characterization of each point in space across a wider array of metrics which you might be interested in. So I think this is nicely summarized by this chart from a review by Christine Tardif. So on the left on this chart, you have a number of alterations or mechanisms at more of the cellular level that you might be interested in. And then at the top, you see different MRI measures such as morphometry, in this case referring to stuff like cortical thickness and surface area, T1, FA, MD, and et cetera. And then the intersection of each of the mechanisms and MR metrics, a square is colored black if the given MR-derived metric is sensitive to a given cellular or a mechanistic phenomena. So this qualitatively illustrates how combining MR modalities may increase your sensitivity to a given mechanism of interest. For example, if you were interested in neurogenesis, by combining morphometry measures as well as mean diffusivity, you increase your range of sensitivity. So it's important to note that this may or may not be desirable for your project. If you're interested at the end of your project, if you want to be able to say something like, you know, this change is reflected of this specific mechanism, then by increasing your range your range of sensitivity to a variety of mechanisms, that's actually a bad thing, right? Then it's harder to say this change is a result of this. But if instead you're interested in, say, generally age-related changes, there you want to be sensitive to a wide array of things happening in the brain at the cellular level. And so in that sense, combining multiple metrics together increases the range of your sensitivity, and that's actually a good thing. So there's been a lot of works recently which have benefited from this combination of multiple MRI metrics. So a couple examples are shown here. On the left, an atlas parcellation from the Human Connectome Project Group, where the inclusion of a range of functional and structural-based indices allowed a semi-automatic demarcation of fine-grained cortical areas. So the takeaway here is combining the different metrics allowed the authors to delineate areas on a finer scale basis than if they only had one piece of information. And on the right, a figure from some work from Jacob Seidlitz, where a combination of structural and diffusion-based indices informed analysis of cortical development and brain organization. So an important note when you're looking at using multimodal MRI is ideally it's better to combine information together at the level of your analysis as opposed to running an independent pipeline for each individual MR modality and then combining at the level of interpretation. So by combining at the level of analysis, you make yourself sensitive and benefit from the covariance across the metrics as opposed to if you were to combine at the level of interpretation. So if we go back to that chart, I'm just pulling out a couple rows from the myelination row in this chart. So myelin is T1 and MD are both sensitive to myelin, but the directionality is opposite. So T1 is positively correlated with myelin and MD inversely correlated. So by combining metrics in your analysis, you open up the sensitivity to this covariation between metrics, which is a benefit. And so in my analysis, to incorporate the multiple metrics together and to become sensitive to that covariance, I turned to a technique called non-negative matrix factorization, which I'll refer to as NMF. So NMF is a matrix decomposition technique which decomposes an input matrix on your left into two matrices shown here as W and H. So the input matrix has dimensions M by N, M rows. The W component matrix has M rows and K columns, where K equals the number of components, and that's selected by the user. And the H weight matrix has as many columns as the input and K rows. So together, the product of W and H describe the input, and so they should reconstruct the input as accurately as possible. And they can be used to describe patterns of variance across both axes of the input matrix. So if you're interested in what are the patterns across the rows, you can look at W, and you can look at different rows which share similar component scores. Conversely, if you're interested in the variance across all the columns, you can look at the H matrix. So a key part of NMF is the non-negative restriction as implied by the name. NMF requires the input to be non-negative, so 0 or above, and outputs as well. So this leads to an additive part-spaced representation, which can help ease interpretation and also is particularly suited to certain MR metrics, which are themselves non-negative. So in the context of a neuroimaging analysis, this analysis may look something like this. In your input, each row may be a voxel and each column a subject. And so if we were to consider analysis of, say, voxel-based morphometry, here each column is a given subject's VBM metrics across a number of voxels. And then the W matrix tells us which voxels share patterns of covariance across those input metrics. And then conversely, H would tell us which subjects share patterns of covariance. So in this way, we have an input where we have voxels and subjects as our dimensions, And with NMF, we can look at patterns of variance across both the voxels and the subject simultaneously. So this approach has been used in a few examples before. One of the earliest examples that I saw, and which actually led to a lot of inspiration for my first project, is matrix factorization of genomic data in an animal model of the hippocampus, where the authors identified output hippocampal clusters showing similar genetic expression. More recently, there's some work by Satiris et al., which looked at matrix factorization in both animal and human data. So what I'm showing you here is some sample output components for NMF on the bottom row, as well as other common decomposition techniques, ICA and PCA on the first two rows. So comparing the two, you can see that the NMF outputs are nicely clustered, but I think an important point here is the NMF, the color bar here is red as positive and blue as negative. And so in each NMF component, you only have positive values, which means you can interpret your output as purely additive, as opposed to having to handle subtractive effects, which you would get in the other decomposition approaches. In this paper, the authors also made use of the weighting matrix to look at how different subjects varied across the different components. So what they did here, which is I think a really useful thing to do, is it was an age-based analysis of VBM data, and they organized their inputs such that each, as you move from left to right across the columns, you're moving from younger to older subjects. So that means when you look at the output weightings here of each component, you can easily visually see that weightings decrease as you move from older, or excuse me, as you move from younger to older. So they used NMF to look at across VBM, and in this matrix, you can visually see, without actually even testing anything yet, that they're outputting components of variance related to age. And then importantly, when looking at the same the corollary outputs for PCA and ICA, it was less obvious, and those age-related patterns were less prominent. And so that gave us support to use matrix factorization in my first project, which was recently published. And as I mentioned before, here I'm looking at microstructural variation specifically in the hippocampus. So I chose the hippocampus not just because it looks cool and it's my favorite brain structure, but because it has some well-documented and important interstructure variations. So on the left, you see an example from our group, which is delineation of the hippocampus subfields according to structural or cytoarchitectonic information. So using this type of information, you can get subfields which show medial to lateral as well as superior-inferior differentiation, but are relatively preserved along the long axis or along the anterior-posterior axis. And then conversely, on the right, you see an image from a review by Strange et al., where they talk about both genetic and functional data pointing to differentiation along the anterior-posterior axis instead. So depending on what type of information you're looking at in the hippocampus, the direction in which you expect to see the spatial variance can vary greatly. And spatial specificity in the hippocampus is especially important because it's a structure that's, as a whole, involved in a wide array of functions and diseases. And so there's an extra benefit to getting that specificity to know which part of the hippocampus is involved in which function, or which part of the hippocampus is most vulnerable in a given disease. So that made the hippocampus a great choice for our first project here. So the data I used was from the Human Connectome project. I used structural and diffusion imaging, specifically T1, T2 weighted image, as well as diffusion. I focused on a set of 329 unrelated subjects from the Human Connectome project with mean age and a female-male split as shown here. And so the metrics I used were the T1 ratio of the T1 over T2 weighted signal, which is a correlate of myelin. And I also used mean diffusivity and fractional anisotropy from the diffusion weighted imaging data. So together, these three metrics gave indices on a voxel by voxel basis of myelin, as well as axonal density and organization in the hippocampus. So then we took that data and built an input matrix that looks like this. As previously shown, each row is a hippocampal voxel, and each column is a subject metric combination. So here, the input is actually, or you can visually see three different chunks according to the three different metrics. And what I want to point out here is the first column of the matrix is subject 1's T1 over T2. The first column of the MD chunk is that same subject 1's MD, and same for FA. And so in that way, it's a bit of an extension to what I showed previously, where instead of voxels by subjects, we actually have this voxel by subject metric combination. And so that means that for a given subject, there's multiple columns in the input, and that means in the output weightings for each subject, you have weightings in each component for each of the metrics. So that is what we use to apply matrix factorization in this project. A big part of any of these analyses is selecting the number of components. So as I mentioned before, the number of components, often referred to as K, is selected by the user. And so this means that if I ask NMF to give me 100 components, it will do so, and then it's my job to say, do these components make sense? So typically what you're working with here is a balance of complexity and accuracy. The more components you have, the more complex your interpretation is, because the more patterns you have to analyze. But at the same time, you don't want to just analyze one or two if that doesn't accurately reconstruct and represent your input. And so I used a combination of this to select number of components as shown in these plots for both the left and right hippocampus. In blue, you see a line that corresponds to the change in reconstruction error. So the blue point at 3 represents the change in the reconstruction error when moving from 2 to 3 components, and so on for each of 3 to 10. Meanwhile, the red line represents a spatial stability coefficient that I developed. So this stability coefficient represents the spatial similarity between output NMF components in 2 splits of data. So a high stability coefficient tells us that the output spatial patterns are replicable within the data set and representative of the vast majority of the subjects, as opposed to being driven by a smaller subset of outliers in some way. And so we want high stability, but we also want to capture as many patterns as possible. So I used these 2 plots to select 4 components as the main point of analysis. Stability is quite high here. And if we look at the change in reconstruction error from 3 to 4, it's quite different than the change when moving from 4 to 5. So when moving from 4 to 5, the change is much less. And so I use that to say that the majority of the variance has been accounted for at 4 components. And given that stability is still high before tailing off, that's what I use to select that level of that granularity for further analysis. And so now I'll show some outputs from that 4 component solution. So if you look at the top left, I have that schematic of the NMF decomposition again, except I'm highlighting the W matrix. So that's voxels by components. And so the variant of NMF we use here has an orthogonality constraint, which actually makes it very easy to obtain an output clustering for each hippocampal voxel using this matrix. We can look at the W matrix and simply say, cluster each row or each voxel according to the component with the highest score. And then we can map those clusters back spatially to either a 3D image on the left or to a slice view on the right, a coronal slice view on the right, and use that to look at the output spatial patterns from this 4 component solution. So I think if you're familiar with hippocampus anatomy, the biggest takeaway, or maybe the first thing you say when you look at this image, is they are subfield-like but not identical, which I think is what I expected going in. So we have 4 components. The largest component is the orange, and it represents, it's concentrated in the center of the hippocampus, and it's I guess most similar to a CA4 dentate gyrus-like subfield. And then we also have components, the pink and the green here, which represent or look like subicular and CA1 areas. But then what we want to do is we want to use the weight matrix shown here on the right to actually tell us what's happening in each of the components. So in the weight matrix, that's 4 components by subject metric weightings, and I've added a little square here to tell you which component each row corresponds to. So if we look at the orange, that's component number 1, we can see where it is in the figure on the left, and we can use the weight matrix to see that on average across all subjects, mean diffusivity is being preferentially added here in comparison to T1 over T2 or FA. So we can say at the group level, MD is higher than T1 over T2 or FA in this orange component. If we move to component 2, we can see instead T1 over T2 is higher than MD or FA in this pink subicular-like component too. But you can also see that in addition to the group level conclusions, there's also considerable variability within each metric and each component. So, okay, at the group level, T1 over T2 is higher than MD. We also see variability within T1 over T2 weightings, suggesting some subjects have higher T1 over T2 than others in this pink component. So the next part we wanted to do is to relate that variability to a set of demographic and cognitive measures. Oh, sorry. We can also relate the findings here to more fine-grained histological analyses. So the first thing to note when looking at this is you're not going to get the level of specificity because of the resolution of MR compared to something like this. So this is an image from a polarized light imaging analysis of the hippocampus from Zaina et al., published in 2017. If you're interested in hippocampus anatomy or cool anatomical techniques, I highly suggest giving that paper a read. So what we see here on the black and white image, this is a transmittance image where darker corresponds to higher myelin and lighter corresponds to lower myelin. And then on the right, the colorful image, the colors and the brightness of the colors denote direction and prominence of fiber orientation. So if we were to focus on the subicular areas, we can see it's darker, so increased myelin, and then we can also see more fibers. And then that lines up with what we see increased T1 over T2 in this pink subicular-like components. And then conversely, in the dentate gyrus-like components, we see increased mean diffusivity, which lines up with sort of the inverse in these patterns here. But as I said, because of the obvious resolution discrepancies here, what you can draw from this is sort of that gross level difference, higher T1 over T2, higher myelination in subicular-like areas versus the center of the hippocampus. Whereas in something like this, you obviously get a much finer grained analysis. And even within something like the subiculum, you can draw conclusions as well. So then we moved on to relating the weightings to a set of demographic and cognitive scores. And for this, we use PLS, or partial least squares. So partial least squares is a multivariate technique which aims to relate two sets of variables together. In this application, PLS relates a set of brain variables, here are NMF weightings, to a set of behavioral data. So here, a set of demographic measures and cognitive tests. PLS outputs latent variables, which can be thought of as patterns describing relationships between the brain and behavior data. Each LV has three outputs of note. There's a singular value, which describes the percentage of covariance explained by a given latent variable. And you also get a score for each brain variable and a score for each behavior variable, denoting how much or how little they contribute to a given pattern. So in our analysis, we identified two significant latent variables for each of the left and right hippocampus. The brain and behavior plots are shown here, but just for conciseness, I'm just going to focus on the first latent variable in the left hippocampus to give you an idea of how to interpret this. So that's shown here alongside those four components. So if we look at the behavior plot here, we see increased age, female gender, and episodic memory. And then on the brain side, we see decreased MD in component 1, 2, and 3, which you can map back here. So that's almost decreased MD throughout most of the hippocampus. So how to interpret this would be sort of a phenotype of older females who perform better on episodic memory tests, showing decreased mean diffusivity throughout most of the hippocampus. And one final thing that we did in this project was compare the spatial stability when using multiple metrics versus when using only one metric. So it's maybe a bit of a boring result in comparison to the hippocampal clusters, but I think it's actually the more transferable output of this project to other analyses. So what I'm showing here is spatial stability. The red line is the same as the plots you saw before. So high stability is a good thing. But then in green, blue, and black, I'm showing spatial stability of output clusters at different granularities when using only T1 over T2, only MB, or only FA. So as you can see, using the multiple metrics together and benefiting from their covariance patterns allows you to draw finer scale components at a more consistent basis. So if you want to get as much spatial specificity, these plots are showing you that there is a benefit to including multiple metrics. So I think that's logical, but that also lines up with previous works where, for example, the Glasser et al. Parcellation, where including multiple metrics allowed them to draw finer scale boundaries. So now I'll come to the conclusions of this work. You can make some conclusions based on the output hippocampal clusters. In terms of microstructure, the hippocampus shows differentiation along both medial lateral and superior inferior axes, and they are subfield-like, but definitely not identical. The combination of NMF plus PLS allowed us to identify brain behavior relationships. And in reading, there's a variety of other works that have also used matrix factorization in analyses. And a common theme that I've found when reading through those and their discussion sections is that NMF is a useful technique for telling you where to look. So in a data-driven fashion, it identifies spatial components where there is relevant variance occurring, and here that manifests through our NMF plus PLS analysis. And then one addition that I would add on top of those works is when considering where to look, the inclusion of multiple metrics allows you to do so at an even finer scale basis. So from that, we move to project 2, where here I aim to investigate microstructural correlates of age and cognitive performance using the Whitehall II imaging substudy data. So now moving to a healthy aging population, the variability on the cognitive performance side becomes of even more interest than previously, where I was working with healthy adults in the Human Connectome project who actually show a low degree of variability across their cognitive scores. So just to illustrate that, I'm showing a plot from previous work from our group of the 8S13 score, which is a score administered to elderly and dementia patients, indicative of cognitive function and dysfunction. So in this score, higher is actually worse. And what I'm showing here is a clustering of different trajectories, longitudinal trajectories across time from the ADNI dataset, where you can actually map out 3 different trajectories. So a maintainer in blue, and then 2 variable decliner groups in green and red. So when moving to a healthy elderly population, in addition to the variability on the microstructure metrics and on the brain side, this becomes of even more interest to see what aging trajectories those variables relate to. So in this analysis, we're using a very unique data set from the Whitehall II imaging sub-study. So the Whitehall II study was started in the UK in the 1980s, and it's a very, very large study, about 10,000 total participants who were civil servants in the British government. And these individuals were followed longitudinally since the 80s, and then in around 2010, a study started where a random selection of them acquired MRI data. So that's the subset we're going to be focusing on now. So we have 398 subjects with demographic info shown here. And we have structural and diffusion weighted imaging, which we'll use to look at microstructural variance. So this is just a depiction of that longitudinal data collection that I mentioned. There are 12 phases, and of interest here, cognitive testing was performed at phases 3, 5, 7, 9, 11, and 12 with an MRI collected at phase 11. And the following cognitive tests were collected at each of those phases, which assess a range of functions, including inductive reasoning, verbal memory, phonemic and semantic fluency, as well as a vocabulary test. And an important and very unique feature of this data set, again, that I want to highlight is the retention rate over such a long period of time. It's around 80%, which I think is remarkable for such a large number of subjects over such a long period of time. So it's a very unique data set in that way. So here we move from just the hippocampus to look at microstructural metrics across the cortex. So we took cortical indices of cortical thickness, surface area, fractional anisotropy, and mean and radial diffusivities, which gives us an input matrix such as this. So here we have each row is a vertex, and we see those chunks again in our columns. And I've added the white lines here just to delineate the different input metric chunks. And similar to that Satiris paper that I mentioned, I've ordered subjects from youngest to oldest. And so visually, you can already see in something like CT, decrease across age from cortical thickness, which is what you would expect. And you can see similar things in the diffusivity metrics as well. So we then apply that, or we input that matrix to NMF, and then using the output weightings, we again look at partial least squares analysis. So here we look at the spatial stability and the change in reconstruction error. Again, this time I've selected K equals 10 as the level of analysis. After 10, there's a large drop-off in spatial stability, and most of the largest changes in reconstruction error have happened. I do note there is an argument, I think, for going a little further on where this blue line plateaus even more, but I made this selection because the drop-off in stability seems to be much higher than the gain in accuracy if we were to move from, say, 10 to 25. So here I'm showing the output spatial components from that 10-component NMF solution. Some general observations throughout are they're relatively bilateral, which is a nice sanity check, I think, and they generally cover non-overlapping regions of the brain, although there is some overlap due to the continuous nature of the output components. And so now I'll move to the result of our PLS analysis, where we took those component weightings and related them to the cognitive intercept and slopes, which we extracted via linear mixed effects modeling of the longitudinal cognitive data. So here I'm showing latent variable number 1, the behavior plot for that latent variable. The colors here are just denoting the intercepts and slopes for pairs of tests. So the same color just means it's corresponding to the same test, either the intercept or the slope. And so what we want to look at here is the brain behavior relationships, but for behavior, it's actually a cognitive trajectory as opposed to just a test at a single time point, which is quite interesting, I think. So we had 3 significant latent variables. I'll go into some detail on 2 of them, although I'll show them each briefly here. The first latent variable accounted for 52% covariance, second for 24%, and then the third for much less, 8%. So I'll be focusing on the first 2 at this point. So latent variable number 1, as shown in the pattern on the right, shows negative correlations across almost all intercept and slopes. So that suggests a pattern of decreased intercepts and slopes across the board, but more generally, it's showing a pattern in which almost all intercept and slope data are correlated. So this is indicative of individuals who either have a lower starting point and steeper decline, or conversely, a higher starting point and slower decline. Here's a histogram of the latent variable 1 behavior scores. So each individual actually has a weighting describing how much they express this pattern. So if you are very high on the right end of that histogram, you show this pattern. Whereas if you are on the low end, you actually show the inverse. So you can use this latent variable and these behavior scores to separate individuals along this axis of high starting point, slower decline versus low starting point and steeper decline. And so you can visually see that if we map individuals' behavior scores versus some of the actual intercept and slope data. So I've selected just 2 tests here just for visualization, and I'm showing you the correlation or just a plot between their behavior score and the slope and intercept of this test. So you can visually see individuals with a high LV1 behavior score have a lower intercept and a lower slope, and the opposite is true for a low LV1 behavior score. And then conversely, for behavior that does not factor into this latent variable, such as the annual recall slope, where the confidence interval spans 0, so we can't make any conclusions about this test in this latent variable, you can see that, okay, perhaps we can pick out relationships, but it's definitely not as clear as the previous slide. And so in conclusion, this first latent variable, the scores here separate those with a high intercept and slower decline from those with a low intercept and steeper decline for most of the tests used in this analysis. So what I haven't shown yet is the brain NMF weights which relate to it. So on the brain side, we get a plot like the one shown here, where for each component and for each metric, we have a bootstrap ratio describing its contribution to this latent variable. The black lines here denote a confidence interval of 99%. So only those bars that cross that black line, I would say, confidently contribute to this latent variable. So instead of looking at this plot, what I'm going to do is just switch to visualizing the actual component maps and color code things as according to positive or negative correlation. But just for illustrative purposes, if we were to look at component 10, what this is showing is that this pattern of low starting point and low decline, and steep decline, is associated with increased diffusivities in component 10 and decreased FA. I think it's easier to visualize this like this here. So I've just mapped the components which play a role, and then I've color-coded each metric according to how it plays into this component. So if you go through each of them, we can say we have decreased thickness and increased diffusivity in component 1, which has focal points in the superior frontal and lateral temporal regions. If you combine, or if you kind of integrate everything all together, we can see that cortical thickness is actually widespread decreases throughout the brain, which is not too surprising when considering age-related decline and decline in cognitive performance. And we see something similar for diffusivities as well. But I'll note, which will come up in the next component, that there is no role of surface area in this latent variable. So then an important point here is, how do common demographic variables factor into this latent variable? So in the PLS analysis, I didn't control for them by including them, but instead what I'm doing here is plotting that LB1 behavior score, describing where you fall on this latent variable with things like age, education, and sex. So in doing this, we can see that individuals scoring high on this latent variable and displaying that low starting point, low decline, are the older and less educated individuals. And we can also see that there's a slight sex effect here as well. Moving on to latent variable 2, here instead we see inverse relationships between most of the intercept and slope pairs. So this is now indicative of individuals who either start high and then have a steep decline, or vice versa. So this LV is instead saying there are individuals who, regardless of their starting point, end up at a similar point in time. And here are the NMF weightings which contributed to this latent variable. So component 1 and the diffusivities come up again, similar to component 7, decreased thickness. So regardless of LV1 or LV2 and kind of the pattern of, or the relationship between intercept and slope, these are two features which seem common to decline, or a steeper decline. However, here we see some surface area effects, where we have increased surface area in component 1 and component 8 relating to that increased starting point, which starts to sound like some sort of reserve mechanism might be at play here. And again, we can look at the age and education effects here. So in this case, there's no relationship between LV2 behavior score and age, so it's not necessarily just separating old from young, but there is, again, an education effect, maybe supporting the idea of something regarding reserve being at play here. And so I have some brief conclusions from this project. So in terms of the component maps, component 1, diffusivity, and component 7, cortical thickness, were common to decreased decline. And then looking at the two patterns of LV1 and LV2, education, which is sometimes a proxy for cognitive reserve, was at play in both of them. So LV1 is more a higher starting point and a potential protective effect of any reserve, whereas LV2 was conversely, regardless of your starting point, a steeper decline. So these are kind of two differing parts of the reserve hypotheses, but this suggests that they may both be at play in this data set, although to varying degrees, because we can say that LV1 was much more prominent in terms of the percentage of covariance explained. So I keep these conclusions a bit brief because there are some definite next steps for this analysis, which is still in progress. So for both latent variable 1 and 2, I talked about the effect of age and education and how they play a role into these latent variables. So currently those weren't accounted for in the slope modeling, but in the linear mixed effect models, if we instead look to account for those things, then that might help that might help remove their effects from the latent variables. And then something else that I'd like to do is, in similar fashion as some previous works, is to describe the components a bit more or in different ways as opposed to just the focal points of the components. So what I'm showing here are two figures from two recent papers where they looked at, in this case, gradient values in different types of, in cortex with different laminar differentiation, and similarly, different metrics within different cytoarchitectonic classes. So in addition to saying component 1 is high in these areas, we could say component 1 is higher in different cytoarchitectonic classes than others, which would help describe them, I think, in a bit more of a concrete fashion. And so that concludes what I wanted to talk about today. There are lots of people to thank, all listed here, and I'll be happy to take any questions.
Thanks, Ray. Great stuff as usual. We have time for questions from anyone. You can type them into me, or there should be a raise hand button somewhere where you're welcome to come and ask a question. I'll start, I guess. So can you take us through kind of maybe a bit of the rationale for not including years of education and age in those initial variables?
Right. So most of it came from trying to focus on just these brain behavior relationships. So there's an argument for it either way, but my thought with not including it here is by limiting the PLS inputs to just the cognitive data, I'm looking at relationships between the NMF weights and those cognitive scores. Whereas if you were to include something like age and sex, the relationship between the brain variables and age versus the relationships between the brain variables and the cognitive scores would be of different magnitude, such that you could expect age to strongly correlate with both brain and behavior more than brain and behavior correlate with themselves. So they're more subtle relationships. So there's some literature that backs this up, but this is also what I've seen in both the Human Connectome Project data and in this data as well. So by not including them, the idea was to try and focus on those subtle relationships versus having the latent variables be driven by age or sex effects. So obviously it's still important to describe the role of age and sex in each of the latent variables. That's what I'm trying to do with something like this, but that's the kind of thinking I had with not including them in the latent variables. I mean, I did start with that, so that's shown here. This is latent variable number 1 with age and education included, and you can actually see this pattern is pretty much the same as the latent variable without them, but you can see here that the relationship with age is much stronger. So by not including them, I wanted to say don't drive the effects, or don't have age and education drive the effects, and then post hoc I'll look at how it plays into them.
Cool. And then maybe could you give some insight on what you think this could give us in terms of, one, I mean, I know I'm leading here because I know you've done this, but looking at function, say with resting state or some other modality like PET, or potentially as a way of examining indices from functional molecular imaging and how they relate to these kind of structural or microstructural measures, and what this kind of technique could possibly give us.
Right. So do you mean at the level of like your input matrix, what are you including?
Yeah.
Right. Great question. So as you may know, Mallar, I'm also working on a project that is kind of like the functional analog of project 1, where instead of microstructural data, you have functional inputs. But what would be even more interesting on top of that is to combine the microstructural analysis, say in project 1, if we were to just focus on the hippocampus, with those functional indices as well. So I'm working on hippocampus functional parcellation on its own without any microstructural data, partially because, well, largely because I'm very new to functional analysis. So before combining things, I wanted to get a handle on it on its own. But yeah, ideally then you would move to including functional metrics, I think, in these analyses as well, because again, the idea is then you can look at the covariance across all the metrics.
I guess one other thing is that I think you touched on it a little bit, but maybe can you talk about some of the trade-offs between using orthogonal components versus components that have the opportunity to overlap?
Right. So I'll go back to this slide here. That's actually a distinction I didn't mention in the first one. So in the first project where you saw hippocampal clusters, we used a variant of matrix factorization that has an orthogonality constraint, which makes the clustering very easy because each voxel is going to clearly have one component score higher than the other. So in the hippocampus analysis, it's not like the component 1 and component 2 scores are close in magnitude. It's clearly component 1 or 2, and that makes the clustering very easy. However, in the second analysis, I did not employ that orthogonality constraints. That allows spatial overlap. So the idea is that although it may make clustering harder, clustering wasn't the goal here, and the goal was more to capture all the variants as accurately as possible. And so in testing some of this, I found that the non-orthogonal fits the data more accurately. And so that's why these components look much more continuous in nature versus the output distinct clusters that I showed for the first analysis. So there's a trade-off there being it's harder to pick a point in space and say this component is in this cluster and this is exactly what's happening it. Instead, it can kind of have like more of a continuous membership in different components. I do note that in this 10 component solution, there's not that much spatial overlap if you focus on kind of the focal points of each component, but that's a trade-off in the sense that it will vary on a case-by-case basis. I think if the goal is clustering, then the orthogonality makes things very easy. But if the goal is more purely on like the brain behavior relationships, then there's an argument for the non-orthogonality, I think.
There's a question here from Katie. Hey Ray, great work. I'm wondering if there are any concerns with including multiple measures from the same subject in the NMF. Is there a way to leverage the repeated measures?
Sure. So do you mean multiple measures as in, subject 1's T1 over T2 and MD is in it, or do you mean something like longitudinal imaging data?
I mean the different metrics.
The different metrics? Okay.
Yeah.
No. So, I mean, it's the factorization doesn't know they're from the same subject. So it's not necessarily biased to grouping those things the same way. So one caveat though is that you can have scaling issues with including different metrics. So in the first project, in both projects, cortical thickness is on a very different scale of magnitude than mean diffusivity. Mean diffusivity values are like 0.00 something. Cortical thickness is like 2. So if you just put the data in without doing anything, you're going to bias it towards the one of larger magnitude because these things are focused on minimizing reconstruction error. So what you have to do is you have to Z-score and standardize the metrics in some way, which is much more complicated than it sounds depending on what you want. But I think that's the main thing to consider. But without imposing any kind of constraints, it doesn't necessarily know that these two columns are from the same subject. So if they look the same, you'll see that. If they don't, you won't.
Does that answer your question, Katie? I think so. All right.
Can I ask something, Mallar?
Yeah, please do.
Ray, I kind of wanted to ask the question the other way. That I was interested in the sort of longitudinal aspect of this. So in the Whitehall study, people get multiple time points of data collection. Are you including all those multiple time points, or is it just sort of a cross-sectional design at this point?
So the cognitive data is longitudinal. So that's included in the PLS by using intercept and slopes from a linear mixed effects model. But the imaging is cross-sectional. So the imaging is just that one time point in green here. So for the NMF, it's just like cross-sectional MRI data. But for the cognitive data, it's longitudinal, and that's captured through mixed effects modeling. Does that answer your question?
Yeah, I think so. I guess to go a little further then, is there a way to to use this model or the results of this in kind of a predictive way, then if you had imaging data from a subject, can you then go back and predict their trajectory based on what you're seeing here?
Yeah, so that's kind of what I want to work towards as well in this project. It's a bit different because it's the cognitive data is retrospective in relation to the imaging, as opposed to the imaging being at the start and then predicting like an input later on. But ideally that would maximize the impact of it, right? Although I've found that is difficult just because the relationships are subtle and prediction is in general difficult.
Right. Well, that's it was great work, Ray. Thanks for the presentation.
Thanks.
Any other questions? All right. Thanks a lot, Ray. That was a great talk.